So when I was invited to speak at this venue, I was trying to think of what would be the most appropriate subject to talk about, and I thought that you would find it interesting to hear about a story. It's an emerging story, it's not a finished story, about work that is at the intersection of physics and biology and mathematics that concerns one of the great intellectual problems in biology, not with biology, but in biology. And if I asked you to make a list of the top few questions in biology that are outstanding, I think we could agree that high on that list would be things like the origin of life and maybe the nature of consciousness. And pretty high up as well is a question that doesn't get quite as much publicity in the social media, let's say, but is a really very deep one, which has to do with the nature of the transitions that took the simplest forms of life that first appeared on Earth out of the primordial soup, and which were presumably like single-cell bacteria, and eventually evolved them to become organisms that are larger in the sense of having more cells, but also more complex in the sense of having more cell types and dividing up life's processes into different specialized cells. And that question is a really interesting one. It's been highlighted by biologists for certainly more than 100 years. But in recent years, many people with a physics and mathematics bent have found this a compelling question because, in fact, when you think about the simplest forms of uni and multicellular life, many of the issues that they have to deal with in their environment are physical ones, like buoyancy and diffusion and mixing. So it's not surprising that people like myself and others are drawn to this question to try to understand the origins of complexity. But in this, what I think must be a pretty diverse and interdisciplinary audience, I'm going to also overlay in my talk a little message, or at least a story, about essentially the tensions that exist between the way physicists and biologists and mathematicians think about scientific questions. And so I hope I pressed the right button here. The subtext is, does biological research require a hypothesis? And I'm sure you already have opinions on this. So I'll weave that into my story in the hopes of stimulating some discussion. I'm not trying to tell you there has to be an answer, but I just want to raise the question. Okay, so the place to begin with this is actually with some data that was first shown to the world by Bell and Moores many years ago, and then represented and discussed by the great late John Bonner at Princeton, who spent most of his 90-some-odd years thinking about the origins of multicellularity. I've shown this graph over many years. It's one of my favorite. It's a log-log plot of the properties of organisms over many different parts of the plant and animal kingdom, showing on the x-axis the number of cells in the body of the organism on a log scale from 1 to 10 to the 15th. On this axis, by some measure of counting that the authors used, the number of distinct types of cells in the body. Okay, so a bacterium has one cell and one cell type, and it sits down here. A highly evolved human, like a director of a center for computational biology, let's say, is about 1/10 of a cubic meter in size, Mike, and has about 10 to the 14th cells. So humans are way up here. Now, if you're a statistical physicist, you might look at this graph and think that there's a straight line somewhere to be drawn. And I would suggest not to do that because it would be pretty noisy. But more importantly, you do see a general trend that as things get larger, they get more complex in the sense of having more cell types. And many people who've been thinking about this problem, I think, would agree that if we're to make any progress in understanding how it is that this complexity emerges, we should be a bit reductionist and look at the lower left-hand corner. Not try to understand everything all at once, but just ask ourselves, if we go from bacteria with single cells, and we start getting organisms with 10 or 100 or 1,000 cells, and then suddenly there are 2 cell types or 3, can we at least try to understand why that happened? What were the driving forces behind it? Can we understand the physics of multicellularity, physics and biology, as it emerges in that kind of way? So that's what I want to talk about, but I'm going to talk about some specific geometrical issues associated with that. And as you can imagine, in biology and in biophysics, anytime you're confronted with these kinds of epic questions, it's natural to find the right kinds of model organisms to use to study. So if we were dealing with genetics, maybe it's the fruit fly, or maybe we should look at C. elegans or yeast or any of those model organisms. For this kind of problem, already more than 100 years ago, people recognized that a particular class of green algae turns out to be tailor-made for understanding these kinds of issues. So these are called the volvocine green algae. They are a distinct set of species that start with an organism called Chlamydomonas. It's a 10-micron across biflagellated photosynthetic eukaryote that you can find in the River Cam, among other places, if you scoop up some water. And it is sort of the origin of a whole set of species with 2 to the n cells. So there's a 4-cell organism, there's an 8 or 16-cell organism called gonium. It goes all the way up to organisms that can have, in round numbers, 50,000 cells and be something like several millimeters across. So these are distinct species. These are not transient accumulations of adhesively sticking organisms. They're distinct species that live their life this way. And one of the interesting features is that all of the organisms to the left of this one, called Volvox, have essentially only one cell type. That is, a cell which at some point in its life is doing vegetative processes like photosynthesis, and then at some point goes through cell division. And it just sort of oscillates back and forth, very much like a bacterium would. Grows, divides, grows, divides. But at this point, one sees the appearance of 2 cell types. There are specialized reproductive cells on the inside, and let's call them galley slaves, which basically just do all the swimming stuck on the outside, somatic cells as they're called. So a natural question is, why did nature put the dividing line at essentially 1,000 cells, or at this characteristic size, which is several hundred microns across? It's a very obvious question to try to ask, but it turns out to be surprisingly difficult to get a precise answer to those evolutionary driving forces. But this set of organisms turns out to be very useful to understand many aspects of this issue of multicellularity. So here's a real-time movie that actually the BBC made in my lab some years ago for a documentary about a river in Scotland. But here you see Volvox in its prime. It's an organism several hundred microns across, so half a millimeter or so. It has 1,000 cells on its surface. Each of them have 2 flagella. Those flagella are like the cilia in your lungs that push fluid around, and they beat in a very characteristic way that makes the organism swim and spin as it swims around a body-fixed axis. And these are the daughter colonies of the organism. And inside of them, you can actually see the next generation. These are the reproductive cells, the grandchildren that will grow up. So it's a beautiful organism. It's been of interest ever since it was discovered by Antony van Leeuwenhoek in 1700 in a famous letter he wrote to the Royal Society. And in fact, historically, there's a very interesting issue that has been discussed ever since the work of Julian Huxley in 1912, written when he was something like 25, which was quite amazing. He wrote this amazingly interesting book called The Individual and the Animal Kingdom, where he talked a lot about organisms like this and asked the question, on what living entity does natural selection act? Is it acting on the organism, or is it acting on the individual inside of it? It's a very interesting discussion. It has to do with what do we call the individual? And it doesn't have to be things that stick together. It could be also separate organisms like bacteria that cooperate together. And there's this amazingly beautiful drawing of Volvox that was made just with a light microscope here. And one of the things he said was there are several different ways you could imagine individuality and organism identity coming together, but he says the other method, aggregation of similar units and subsequent division of labor among them that open to life the full resources of the second grade of individuality. In some colony like Volvox, there once lay hidden the secret of the body and the mind of man. It's a very profound statement, so I feel justified in using this as a model organism if Julian Huxley says that about it. Okay, but one thing I will say is that actually over the last 20 years or so, it's become clear that these green algae are actually interesting on many levels for studies in physics and biology and in fluid mechanics. So for instance, I'll tell you about this multicellularity issue today. One can discover fascinating hydrodynamic interactions between these spinning organisms that result in things called hydrodynamic bound states, which are fascinating dynamical systems driven by fluid-mediated interactions between the organisms. Because they're so large, half a millimeter is huge if you're doing microscopy, and their swimming is so steady, it's possible to measure the fluid mechanical flows driven by those 2,000 flagella with great accuracy, and to test microscopic theories that link the beating of all of those flagella to the large-scale flows around them. There are some very interesting issues about how the fluid flow driven by these organisms moves around tracer particles that may be inside a suspension, leading to unusual kinds of statistics of the displacements of particles that instead of looking like ordinary Brownian motion, the system has long exponential tails. But one of the most important issues that has been studied, which is very important for the human condition, is the synchronization of those flagella, which beat very much like the synchronization that occurs in your respiratory system as mucus is moved around by cilia. And there's also a very interesting question of how organisms like this, which have 1,000 cells and no central nervous system, manage to steer toward the light. They're all photosynthetic. They have to go to the light to do their work. And yet, without a central nervous system, how do they coordinate those 1,000 cells? It's a fundamental question about the emergence of multicellularity. And then more recently, as I'll mention a little today, there are issues about tissue folding that come up when these daughter colonies are formed. They're actually formed initially inside out, and they turn themselves right side out by a process called eversion. It's literally turning a sphere inside out. So it's a kind of Swiss Army knife of a system where you can study many interesting physical problems. Now, Huxley himself, but more recently, Thibaut Brunet and Nicole King, then at Berkeley, pointed out a very interesting connection between 3 different classes of organisms, which turns out to be very motivational for what I want to talk about today. So the connection exists between the organisms known as choanoflagellates, sponges, and the green algae. So choanoflagellates are uni and multicellular organisms that are the closest living relatives of animals. So if you think of the tree of life with the branch that is animals, at the very base of that branch are sponges, plus or minus a small argument, but let's just say sponges. And just to the right or left are choanoflagellates. They're very similar to the filter-feeding cells inside sponges, but they are a distinct species. So they're our closest living relatives of animals. And one of the things that Brunet and King pointed out is when you look at the structure of choanoflagellates, they have a cell body. They have a single flagellum, which they use to filter feed from the fluid around them. And then they have a collar of microvilli. They look like a badminton shuttlecock. And that collar of microvilli is what does the filter feeding. And it turns out when these organisms divide and sometimes form multicellular structures, they form very interesting geometrical structures, sometimes very regular rosette-like structures, by excreting a polymeric matrix known as the extracellular matrix from them. And this glue sort of holds them together in this regular geometry. And it tends to be this radial, spherically symmetric geometry. Now, in sponges, in the early development of sponges, the embryonic form actually ends up forming a structure which in cross-section looks very similar to that you see in the green algae, which are also held together by an extracellular matrix, which is exuded by the cells. So there's this common sort of geometric structure of spherical aggregates held together by an extracellular matrix, which Brunet and King said was an interesting connection. And when I read this and I thought about it in the context of the work that we had been doing over the years, I realized that there was actually a very general question that one could ask in biology in light of this. And that is, how do cells make structures external to themselves in some robust and accurate way? They're excreting polymers. They are self-assembling into some sort of matrix that holds everyone together in a regular fashion. But they don't have any arms. They don't have any eyes. They don't really have a brain. And yet somehow this manages to work. And I thought this was a really interesting question, and it sort of framed what we wanted to do. So you can line these up this way and say, OK, we know about the green algae. Volvox has 1,000 cells that are actually mounted right at the surface of this transparent extracellular matrix. But it's a complex 3-dimensional structure with various kinds of levels of density and structure, which is all a consequence of a self-organization process of those polymers. I mentioned the choanoflagellates. If you look at the structure of sponges in cross-section, not kitchen sponges, marine sponges, you'll often see in the more complex forms spherical chambers with filter-feeding cells that are like these choanoflagellates, again surrounded in this gray by this extracellular matrix that's exuded by the cells. So in fact, some years ago, I interviewed for a grant application in London in front of a very intimidating panel of scientists. And I sort of proposed this, and I said, we're at the early stages of seeing this is a really interesting question, and wouldn't it be interesting to characterize the growth dynamics of these extracellular matrices and try to understand how this regular geometry appears? And one of the members of the panel said to me, yes, but what is your hypothesis about the mechanism by which this happens? And I said, well, I don't have a hypothesis yet because I haven't done the experiments. I'll do the experiments and we'll make a measurement and then we'll see. And she looked me in the eye and she said, but how do you know what to look for if you don't have a hypothesis? At which point I realized I was not going to get the grant. and I have to say, I usually am pretty good on my feet, but I was totally speechless because I hadn't encountered that kind of question before. So I went home and I looked up in the dictionary the definition of the scientific method. And okay, it's the Oxford English Dictionary. There isn't the Cambridge one, but anyway. And it's a systematic, it's a process involving systematic observation, measurement, and experimentation, induction and the formulation of a hypothesis, and then the testing of that. In other words, you can see it as a kind of circle with these various stages. And I just always thought you start with the experiments and you formulate the hypothesis, but apparently there's another point of view, which is that first you make the hypothesis and then you make the measurements, and I was quite confused. Some years ago, I came across the fact that there is a tension between these two disciplines, and it was in the context of how you write scientific papers. Is a mathematical result a result? And it turns out there are differing opinions on this. Some people believe that the experiments are the result, and the interpretation is for the discussion or the supplemental material. So I decided to write this essay, and I did achieve the world's shortest abstract so far. And I'm proud to say this paper's been read 27,000 times, but I have no idea whether it made any difference. But we'll see. Okay, so the first of my three halves of the talk is about the cellular neighborhoods in Volvox and what we can learn by some simple experimentation. So if you go back to Huxley's beautiful picture in the frontispiece of his book, you'll notice that around every one of these somatic cells, there is a polygonal neighborhood identified. And that was hand-drawn by either him or someone he trusted. And it's actually the start of a very interesting discussion, because if you take a very modern view, a beautiful microscopy by Rogelio Moreno, which you can find on the web, and you look very closely under DIC imaging of Volvox, and then you zoom in, what you can see is that those compartment boundaries actually are visible. So these are denser regions of this polymeric matrix that show up as polygonal boundaries around each cell. You can think of that as its neighborhood. Now, if you're just making a drawing, it's one thing, but you'd like to be able to quantify this better. And thanks to my brilliant PhD student, Anand Srinivasan, who's an expert at, among other things, image processing, you can go inside and do some machine learning to actually get a really accurate description of where these compartments are. And what you see is that, well, most of them have 6 sides, some have 5 sides, but there's a broad distribution of the areas associated with each one of them. Yet this whole thing is spherical, and it grows as a sphere. And that's the most elementary question. How does it do that? There's all this noise, essentially, in the geometry of the neighborhoods, yet the whole thing grows as a sphere. Okay, so as I said before, it's a really complex problem. All of those polymers are exuded from the cells, and they self-organize into a structure which has these compartments, but it has other zones in which the daughter colonies grow up, many different microstructures, all self-assembled. And I can't answer for you how that happens, but I can start to understand a little about the geometry. So the life cycle of an organism like Volvox is a very exciting 2-day life. If you think of it as a plant, it needs both light and dark to grow. So in the morning of the first day, like in Genesis, when the light is turned on, this organism, let's say, is about to hatch its daughter colonies. They dissolve their way out of the mother, which then sadly dies. And these daughter colonies have inside of them germ cells, which are the cells that will grow up to be the next generation. In the first day, these germ cells grow very large and then rapidly go through 9 or 10 rounds of binary division to produce the 2 to the 9 or 2 to the 10 cells of that next generation. And those daughter colonies, which also have their own germ cells, are initially formed inside out. That is to say, the poles of the cells from which the flagella will emerge are facing in. Now, I don't know about you, but if I tried to swim and my arms were facing in, that wouldn't work. Right? So nature fixes this by a process called inversion, in which each of those spheres develops a hole at the top, and it turns itself inside out, and then the flagella point outward. And the rest of the life cycle consists of the expansion of the extracellular matrix from that raft of cells to produce the ever larger organism. Okay, so some years ago, we were happily studying by light sheet microscopy two interesting aspects of this problem. One is inversion. So here you see a movie taking about 45 minutes of the process of inversion of one of these colonies. It's quite amazing, I think. So it's a sphere that turns itself inside out by a wave of cell-shape changes that create bends that ultimately invert the organism. But at the same time, it was possible to hold the entire colony. That's a zoom of one of these daughter colonies. And you can see over the course of several hours how the colony is growing. And there's some noise, but it stays spherical. So we were in the midst of continuing to gather lots of data on the geometry, but we weren't quite sure exactly what was the best way to analyze it, which is why the biologists on that panel didn't like what I had to say, but that's okay. And then in the middle of the pandemic, I got an email, a very important email from a group of physicists and biologists at Georgia Tech. And the graduate student who emailed me, Tom Day, basically said, we've been studying a particular form of yeast, which I'll tell you about in a second, and we've come to some interesting conclusions. By any chance, do you have three-dimensional images of Volvox, which we could compare to see if there's any similarity? And I said, yes, we do. Funny you should ask. Now, what was the group of Ratcliffe and others at Georgia Tech interested in? An organism called snowflake yeast. So ordinary budding yeast is something that you grow up in suspension. It's thought of as a unicellular organism. That's the way everyone is familiar with it. But the experiments that Ratcliffe did prior to moving to Georgia Tech with Travisano and others in Minnesota was the following. Take yeast, grow it up in solution, centrifuge it for some period of time so that everything, or most everything, settles to the bottom. Take the bottom few percent of solution, grow that up in a new test tube, but throw away the rest, and just keep doing that. Scores of times, like 50 or 100. And what you discover at the end is that you no longer have unicellular yeast. You have what is known as snowflake yeast, which is a ramified, random object, which is not just a transient adhesive clump of things, because when this organism goes through cell division, it produces a multicellular progeny. It is a remarkable experiment. They turned on a multicellular life cycle by selecting for something which happened to be sedimentation. Larger things settle faster. And they had been studying the geometry of these. The physicists among them had been looking at this as a kind of random packing problem, similar to what you find in granular physics. And they had discovered an interesting distribution of the volumes that you could assign to each of these cells. And that was what they were asking us to check. So the way they did this, which is a standard thing in physics, is to say, consider a collection of cells, look at the cell centers, construct the perpendicular bisecting planes of the lines connecting individual cells, and then you will construct around each cell a polyhedron whose volume you can measure. And then you ask yourself for one of these structures, what is the distribution of the volumes around each cell? If it was a perfectly regular structure, they'd all have the same volume, but they don't. Now we can do the same thing. That's what we did immediately from our data. We can take the centers of every one of these cells, construct the perpendicular bisectors, and thereby construct what are known as Voronoi polygons in the plane. And the shocking thing that we and they found, rather surprisingly, is that when you graph the probability distribution of those volumes, normalized by the volume of the cell, whether it's snowflake yeast or Volvox, it's very accurately a distribution called a gamma distribution. So if V is the volume and VC is the volume of the cell, then in rough units, it's x to the k minus 1, e to the minus kx, where x is just a rescaled volume. Now, if you think about this as a physicist would, you might say, well, these are all random processes. I add together a bunch of random processes. Why don't I get a Gaussian by the central limit theorem? And that's an interesting question, but it's definitely not a Gaussian. It's a highly skewed and very broad distribution. So the question is, where on earth does this come from? And we have some ideas, which I'm going to tell you about briefly, but we're still working on it, which is why this is. Yes.
When you do the image processing, you have the volume of the polyhedron, right? So it's in cubic microns or something like this. So what exactly is VC?
So VC in the case of snowflake yeast is the volume, the three-dimensional volume of each of the cells. Okay, or the average. Right?
Okay.
In the case of Volvox, we're talking about two-dimensional polygons, and we're looking at the projected size of the somatic cell. So you have a somatic cell surrounded by a compartment, basically, and it's that. So the minimum size that the neighborhood could be would be the cell size.
So the size of the cell is included into this compartment, right?
Yes, yes.
Okay, and roughly by how much larger is volume of the compartment to the volume of the cells?
Well, you can just see, right? We're talking 3 times, 4 times.
Okay.
Yeah. Yeah. Okay, so it's also weird because snowflake yeast appeared on Earth yesterday. Okay, last Thursday. But Volvox has been around 500 million years. They have utterly nothing to do with each other when it comes to the method of reproduction or anything like that, yet they share this. So there's something deep going on there. Okay, so we thought about this for a long time, and then with Anand, who's a brilliant mathematician, we realized that there's a branch of pure mathematics that has been thinking about exactly this question for a long period of time, and we just hadn't known at all about it. So the idea is take a surface, a manifold if you're a mathematician, and throw down a bunch of points randomly, and then construct the Voronoi tessellation associated with those points, like this. And what you discover is very robustly, you get gamma distributions out of this. It's just sort of natural, and I'll give you a trivial little derivation of why that is natural. And it seems to be extremely robust. It's sort of independent of dimension. And therefore, there's something rather geometrically simple going on here, it seems. Of course, you have to ask, why are the cells randomly being placed down? That's a sort of separate question. And in fact, the problem here is that the matrix we're talking about is produced by the very cells whose positions we're talking about. So the cells are like the template and the source of the same material. So it's a very weird problem to think about. But anyway, this seems to be what's going on. So you know the story about the spherical cow? Yes? Right? Yeah. Physicist is asked by a farmer, I'm having trouble with my cows not producing much milk. The physicist comes back a few days later and says, well, consider a spherical cow, and we'll start from there. All right, so I want you to consider a linear Volvox and ask yourself, or let's consider the following problem. We're just placing points on the line where the spaces between the points are occupied by this extracellular matrix. So let me make the following statement. If it were true that the amount of material exuded is distributed as a Poisson process like this, exponentially in the length of material, and I consider two neighbors which have produced X amount and Y amount, and I want to make a Voronoi tessellation, I have to find the division line of the sum of the two lengths. And so if I add two random variables together, there's a rule in probability that says I convolve their probability distributions as such. So if I start with that distribution, I will end up with this distribution, z e to the minus lambda z for the sum. And the second thing you need to know in probability is that if I multiply a random variable by a constant, then there's a rule for how the probability distribution evolves. And if you solve that, you'll discover that the Voronoi tessellation of this exponentially distributed process is exactly a gamma distribution with gamma equals, with k rather, equals to 2. And when you measure the Volvox distribution, it's like 2.3. So there's something just about trivial, it seems, about adding up exponentially distributed random numbers, and that gives us this. Okay, now I'm going to show you that there's a very interesting precedent in studies of protein transcription some years ago in other organisms that suggests that it's rather natural that proteins that are produced by cells are produced in bursts, very tightly controlled bursts with exponential distributions of amount of protein produced. And it generally is the case if you produce B bursts in this way, you'll end up with a gamma distribution where k is 2b. And when b is much, much greater than 1, you produce a lot of bursts, a lot of very small bursts, you will get the central limit theorem. You'll get Gaussians. But when you produce a very small number of bursts, you get something very different. And the experiments that were done, and very clever experiments in E. coli by several groups, basically looked at the number of bursts per cell cycle in this particular organism, where they fluorescently labeled the proteins that were coming out, and they stuck to the cell membrane so they could just watch the fluorescence in the cell membrane. They observed a typical burst number in this particular system of a little over 1, and the probability distribution of the number of molecules produced was a beautiful exponential. So that suggests one possible explanation of what's going on here, is that the extracellular matrix components are being produced in a bursty manner that gives these exponential distributions, and that's what gives rise naturally to the gamma distributions. But there's another, sorry, just to make it sort of precise, what we're saying here is, essentially using the central dogma of molecular biology, we have the gene producing mRNA, and if the rate of degradation of the mRNA is really fast compared to the rate of degradation of the protein that's produced, then you'll have this bursty dynamics. So the hypothesis here, actually we do have a hypothesis now, is that it's actually bursty dynamics of these extracellular matrix components that could be the origin of this exponential distribution. And the really amazing thing is that if you take the measurements of these compartment boundaries, and then you make a Voronoi tessellation, which makes no reference at all to where the boundaries are, and you overlay the two, they are remarkably close. Now, that's a separate issue from this one, but it says there's something deep in biology about the way space is divided up. It's almost a beautiful Voronoi tessellation, and we usually think of that as just a mathematical construct. So there's a deep question there for further study. Now, there's another point of view that may be useful here, which is to think of this problem more from an energetic point of view. And some years ago, there was a beautiful piece of work in Neurophysics Letters that proposed the idea of what is now called the Voronoi liquid. So you imagine a world in which you divide up space by Voronoi tessellations, and you say to yourself the following. Suppose the centers from which I construct the Voronoi tessellation are positions xi, so there's a set of such points, and I lay those down in space, and then I construct neighborhoods around each of them where there's an energy functional, which is essentially the second moment of mass around those centers of mass. So it's just a variance of mass around the center. Now, what you can show is that this kind of energy structure is essentially the small strain limit of deformations of regular n-gons from their regular n-gons centered at xi. And if you minimize over these domain shapes, then what you have is a kind of energy functional for the positions of the centers. And you can use that as an underlying energy in statistical physics, where the probability distribution of such occurrences is just the Boltzmann factor with a temperature-like variable. And when you do that, you discover that at very high temperatures, so beta, the inverse temperature goes to 0, you have a random distribution of structures, which is essentially a maximum entropy configuration, which is very distinct from what you find at 0 temperature when you get a kind of regular crystalline packing. So this is like the one limit is essentially the k equals 1 limit, and the other the opposite. So here we have something energetically which allows us to tune between gamma distributions and more crystalline distributions. And the next step is to try to figure out which, if either of these, is the one that governs what happens in these systems. And the only way to make progress is to have some labeling of these partitions where we can understand where they come from. And in recent work that we've been doing with a group in Bielefeld, Germany, under Armin Hallmann, we think there's a way forward. So this group has done a remarkable genetic engineering job of creating fluorescently labeled strains of Volvox where the proteins that actually define the compartment boundaries light up fluorescently. So you can just see them and quantify the underlying microstructure directly. You can see the somatic cells by their autofluorescence. You can see the compartment boundaries by this ferrophorin 2. So this allows us, we think, in the future, to try to get a handle on which of these two points of view might be operating here. Is it about the exponential distribution of material, or is it about the elastic strains, or maybe some combination of the two? So that's where we are on that, but I hope this has given you some idea of how useful this organism can be in trying to answer these questions, and also the geometric issues that come up. Okay, so now I want to turn to a related problem involving a very interesting newly discovered species of choanoflagellates, where some of the same kinds of questions come up. Okay, so that same group of Nicole King and Thibaut Brunet, with a number of other collaborators, back in 2019 made a remarkable discovery of a new species of choanoflagellate. This was on an island in the Caribbean, so they have a very nice field trip to make this discovery. So I remind you of the structure of individual cells of choanoflagellates: cell body, microvilli, collar, and flagellum. This organism, called Choanica flexa, is a raft of cells with no extracellular matrix, but held together by adhesive contacts between the microvilli tips. And so here's a picture of this raft of cells. If you're close enough, you can see the flagella undulating. It kind of oscillates. It's a sort of raft of cells with some elasticity. And the amazing thing about it is that it can exist in two different forms, which can interconvert by changing the light shining on the organism or various chemicals. In one form, the cells are rather far apart, and in another form, they're very closely packed. But more importantly, in one form, it's a bowl shaped like this with flagella pointing in, and the other one is a bowl shaped with the opposite curvature and the flagella pointing out. And one of them swims very well and doesn't filter feed much, and the other filter feeds very well and doesn't swim much. So it's a remarkable sort of bistable shape that nature has engineered. And if you look very closely at the right kind of microscopy, you can see the individual cells, and you can see all these microvilli sticking together. And in the right plane, those microvilli define polygonal regions, sort of reminiscent of the compartments in Volvox. And what we understand from their amazing work is that the microvilli that are sticking together actually have a kind of bistable structure. They have a ring of contractile actin-myosin system that, in cinching together, flares the angle with which the microvilli spread, and the interconversions between these two shapes are driven by that. So when you think about trying to make some sort of mechanical model, some description of where this comes from, one way to do it is the following: to say, look, I want to create a raft of cells that has the possibility to be curved. And if you think at all about a soccer ball, you'll know that you have to have some pentagons and some hexagons to make it work. If it's just a bunch of hexagons in a raft, you can't really bend it very well. So mathematically, the way to do that is to take a bunch of spheres and assign them to the vertices of what's called a geodesic icosahedron, which is just a projection of the vertices of an icosahedron onto a sphere. So you have a raft of cells, which has some distribution of pentagons and hexagons. And then the simplest model is just to construct elastic springs that represent these microvilli that connect every cell via an intermediate connection to another cell with these two microvilli here. And you can assign an elastic energy to describe deformations of these angles from their preferred values, and try to construct some mathematics of the interconversions. So one thing that you find is that you can rather easily explain the differences between the swimming speeds of these colonies relative to the single-cell swimming speeds as a function of the curvature that you apply to the system as you go from flagella out to flagella in, just as a consequence of the sort of porosity of this network of spheres. But the most important thing that you discover is that quite naturally, as you change these opening angles of the microvilli, you can make the system convert from flagella out to flagella in, going through this kind of sequence of shape transformations, very much like what you see experimentally. Now, I have to say that when we were starting to think about writing this up for publication, I was a bit confused, because I thought, you know, we built a model which was based on just Hookean linear elasticity to describe the springy connections between all these cells. And then somehow out of this, we got this very nonlinear process where you could flip from one stable bowl-like configuration to another. And I couldn't really intuitively understand where this bistability came from. And it was a bit of a puzzle. And then I remembered a beautiful paper I had read back when I met Mike many years ago. It's a paper by Sung and Nelson, 1988. They were interested in topological defects in what they called membranes with crystalline order. So it's basically just a consider a network of beads and springs where you have, say, originally a bunch of hexagons, and you introduce a single pentagon somewhere. Not surprisingly, that makes the system buckle into a kind of curved shape in the same way that the soccer ball would. And if you were to put a heptagon in there with 7 sides, it turns out to make a saddle shape. And I went through their analysis over and over again, kept trying to think, where is the biostability in the problem? And at some point in the mathematics, they say, well, consider a small perturbation of the shape away from flat. Let's try to calculate the energetics. And then I realized that there was nothing to say I should make the perturbation go up or down. I would get exactly the same result. And in fact, I went out and I bought a molecular modeling kit to try to understand this. But before I did that, I looked at a picture of gonium, which is the 16-cell organism on that lineage of green algae that I told you about. Here's a beautiful image from the web of gonium. And if you look very carefully, you'll actually see that this cell has 7 neighbors. So it's a living example of the heptagonal defect that Sung and Nelson were talking about. And in our lab, we studied gonium. Hélène de Malaprade, my postdoc some years ago, made this very beautiful movie of gonium as it swims around. And as it swims, you can see it's got a saddle shape. So it's just naturally doing that. So I went out and I bought a molecular modeling kit, and I made one with a few defects in it, pentagons. And I put it on the floor of my office, and I took this picture, and I said, it works, right? I could just take the model, and I could push it, and it would naturally, eventually flip to the other side. So the biostability is a consequence of the presence of the pentagons. So we then said, back to Brunet and Larson, can you measure, from your confocal imaging, the number of pentagons in the system? So they went carefully, and if you take the right plane and you color code things, you can see there are a whole bunch of pentagons in this packing geometry. And in fact, something like 22% of all of the polygons in the system are pentagons. So the biostability is a consequence of the way these have packed together. Okay, so the last thing I want to tell you about is how this connects a bit to the extracellular matrix of Volvox and its mechanical properties. So I told you already about the life cycle of the organism. And after this inversion process, the second day is just spent adding extracellular matrix as time goes on. Now, at this point in the life cycle, you can apply a technique that's well known in the field, which is basically to mechanically shear the colonies, essentially by stirring up the fluid in which they are. And if you do that vigorously enough, you'll break up the colonies. And what we discovered is that when you do so, you end up with pieces of the extracellular matrix, which in these still views, you can see adopt a kind of curled up shape, a bit like seed pods. And here's a video, it's a high-speed video. The oscillations you see are the beating of the flagella, forward and back. But the structure is this seed pod shape. We thought, where is this coming from? What does this mean? And it turns out, after a fashion, you can do 2 things. The first is that you can use the light sheet imaging of the entire colony to identify where all the pentagons and heptagons are. And you discover that out of those 1,000 cells, about 200 of them are pentagons, a fewer are heptagons, so there's a net number of pentagons, and the rest are hexagons. So there are a lot of these topological defects in the system, which would naturally tend to make it curl like the soccer ball or the football. And an asymptotic analysis that was done by another of my PhD students, Marco Vona, closely motivated by the experiments I'm showing you here by Jane Chewy, suggests that the energy of these kinds of systems can be basically written in a form that's reminiscent of something that was proposed many years ago to describe the energetics of things like red blood cells. So it's a surface integral of the deviation of the local mean curvature from some preferred value h0 with some elastic modulus in the front. So the system would naturally like to have a mean curvature which is equal to some preferred value, like a bimetallic strip likes to bend, and you pay a price quadratically to deviate from that. So there's a preferred curvature built into the system as a consequence of the way this ECM grows. And if you follow that energy through, you can do the following calculation that Marco has done. You can imagine taking a sphere and chopping pieces out of it, and then asking for it to relax to the new shape that it might find as its energy minimizer. And if you just chop the top pole off, it doesn't change much. But once you get down to a small segment like this, you naturally find that it wraps up into this seed pod-like shape. So we have a chance of measuring the intrinsic curvature of these pieces of the extracellular matrix. Okay, so the final comment I want to make is it possible perhaps to make some progress in understanding the more complicated sort of holy grail of the problem, which are these sponges? How does this complex microstructure develop? A sponge starts its life as a sphere with the mathematical topological genus, not the biological genus, of 0. And by the time you encounter the mature sponge, it has a genus of, I don't know, 12,009. Right? How does it produce this complex microfluidic geometry in some regular fashion? Nobody knows. But there are two things you can say. One is that from beautiful work of Sally Leys and others over the years, if you look at the microstructure of these spherical chambers lined with filter-feeding cells called choanocytes, which are very similar to choanoflagellate cells, and you look very carefully at the network of packing, you'll see those pentagons again, which suggests to me at least that could be a self-organization process of these curled up domains, very much like what we see with C. flexa. But the second thing is the only way we're going to make progress really is to have some way of looking inside a sponge that's living to understand its microstructure directly. And this is ongoing work with Kenji Kikuchi and Takuji Ishikawa in Sendai, in which at least this is the first indication it might be possible by suitable imaging of fluorescently labeled parts of sponges, either fluorescently labeled microparticles or appropriate live stains, you can actually start to see the microstructure of all of these chambers together as you scan through the organism. And we hope this will give us some way of untangling the developmental process that leads to this. So when we started looking for interesting aspects of extracellular matrices, I had no idea that we'd find gamma distributions and all the things that we found. So I think it's fair to say, at least in my limited experience, does biophysical research require a hypothesis? No. It may be useful. I don't think it's required. And this is an article that we're struggling to write with Kyriakos Leptos, who's a senior research associate in my group, and Wilson Poon at University of Edinburgh, who's thought a lot about this issue. But more importantly, in encountering the difficulties of doing interdisciplinary research, I want to just quote from two things a referee said about one of our papers. The paper was published, so I don't have any qualms here. The data should be described and the inferences drawn and the modeling relegated to its proper place as a quantitative verification of the inferences that can be made directly from the data. And later, instead, the author should let the data speak for itself and postpone heavier theoretical analysis for later, perhaps in the discussion. Obviously, I don't agree with that. But I think it raises a very interesting question, which is worth discussing, which is, if so, what language does the data speak? And the answer is it speaks differently to different people. We see different things in it, and there's no one-size-fits-all to these kinds of approaches. And I hope today I've given you a little insight into one way of looking at them, which is motivated mostly by physics and mathematics, but clearly with care for the biology. So thank you very much. I just wanted to dedicate this to the memory of John Kessler, my longstanding colleague at the University of Arizona, who was very motivational in getting us all interested in these problems. Thank you.